when she got ill, went home to England and died a few years before the Nobel Prize. She's now recognized by the United Kingdom as the top woman science world award every year is the Rosalind Franklin Award, and everybody recognizes her key contribution today. But and she did publish a paper, a subsequent paper after this, that talked about the chemistry. So she was known. I mean, her reputation was solid. In fact, she got a very high-level job. I don't know if it was at Cambridge or Oxford that she had until she passed away. So she was known, but she didn't at this point didn't get the Nobel Prize in the early '60s because she was no longer there. Okay. So the paper just talks about kind of the chemistry of their hypothesis that DNA is a double helix. Okay. The backbone is a sugar phosphate backbone shown here as a ribbon. Remember, the phosphates have negative charges, so that's a repulsive force. We'll have to pull it apart. What holds the two strands together, and shown by these rungs in the like rungs of a ladder, are the complementary base pairs that hydrogen bond together and hold the molecule together. Now we know hydrogen bonds are individually weak, but we're talking about a molecule that has 100 million of these, and that can stabilize the molecule, stabilize its integrity. Okay. So this is their proposal. Now, I said the goal was to describe the 3D structure of DNA so it could account for two things. One is how is the code embedded in this molecule, and how can it be copied before cell division. Okay? Now, in terms of the next slide, we'll talk about how the code's embedded. Okay? But I just point out the last sentence of the paper before the acknowledgement here. It says, it has not escaped our notice that the specific pairing we have postulated, that is the A with T and the G with C, okay, immediately suggests a possible copying mechanism for the genetic material. Okay? So their structure does satisfy both questions that led this scientific quest. It will account for how the molecule is embedded in the code and how it's copied. Now, in terms of how the molecule, the information is embedded, I said that wrong, that's what I meant. We're going to look at these different versions of the double helix here. We'll look at this one here. We'll look at this one in a second, more detail. But the ribbon here represents the sugar phosphate backbone. Here it is in gray here. Sugar, phosphate, sugar, phosphate, sugar, phosphate. Phosphates have a negative charge, it's not shown. It's the sugar phosphate of the complementary sequence. And then you can see how the hydrogen bonds hold the complementary base pairs together. This shows all combinations B, C, and G. 3 bonds between G, C, 2 between A and T. Okay. Well, how is the code embedded in this molecule? It's a sequence of nucleotides along one strand. The alphabet is only 4 letters, but any sequence, you have an unlimited number of possible sequences, just like our language has 26 letters. DNA is a linear language like the English language. We can always coin a new word by changing the spelling. We change the spelling, it means we have that means one thing, that means another, that means another. Okay, change the spelling, it has a new meaning. Change the sequence of this 4-letter alphabet, and it spells hemoglobin, or it spells a muscle protein, or it spells an antibody. Okay? So any spelling is possible, embedded in one of the 2 strands. What about the replication? Well, that's where the complementary code comes in. Okay? We'll see how the base pairs imply a mechanism for copying. Okay? Now, a couple of things about this molecule we want to point out, because there's some seem like trivial details that are important to understand, things like replication, transcription, and PCR that we're doing next week, polymerase chain reaction. One is that the DNA molecule, okay, this double helix, has an anti-parallel structure. What does that mean? Well, if you look at the 2 complementary strands, and I think the best way to see this is look at this version here. Look at the shape of the sugars, this 5-carbon deoxyribose. If you look at the oxygen in the ring as sort of a place to gaze, notice that like an arrowhead is pointing up, this one's pointing down. So the 2 strands are kind of head to toe. That's what we mean by anti-parallel. Okay? Anti-parallel. We refer to that by calling one end the 5' end, the other end of that one strand of 3' end. Because the anti-parallel, 3 pairs to 5 and 5 to 3. Now where do those numbers come from? Oops. Just keep me on the, oh, this one didn't work, sorry. Too bad. Here's where the numbers come from. If you look, we've numbered the sugars, the carbons of the sugar. They're numbered 1', 2'. Of course, as we know from our understanding of the shorthand drawn structures of organic molecules, since they're carbon rich, usually the carbons are left out. And if there's another element in a ring or a chain, they always indicate, in this case, oxygen. So these are the numbered carbons of the sugar. There's carbon 1, 2, 3, 4, 5, or 1 through 5 prime. The 1' carbon is always attached to the base, and you go clockwise. 1, 2, 3, 4, 5. Now, people don't wear watches anymore, but you know a clock. Clockwise. Yeah, just in case everybody looks at their phone. I see a guy skateboarding off campus with his phone, and they go, that's smart. Cars, I don't know where my cars. Curbs, who cares? Right. But if you look at the 3' end here, it's the 3' carbon of the sugar. It's kind of open to the end, and the 5' is embedded in the chain. Look at the top of this. So if this is 3', this has to be 5'. Look at this end of the chain. Carbon 1, 2, 3, 4, 5's at the top, with the phosphates attached. And then because of anti-parallel, this is the 5' end. Notice at this end, it's 3' carbon. Here at the end, this 5' is embedded in the chain. And since this is down here's the 5' end, you see this, go find the nitrogen connection, the bond between the sugar and the base, go clockwise 1, 2, 3, 4, 5. It's at that end. So it seems like you're so like, really? You got to tell me that? Well, it turns out that a lot of the biochemical processes like transcription or replication, transcription can only go in one direction. Okay? And when you look at a DNA sequence in a database, okay, they only report one sequence of the DNA because we know the complementary sequence, right? So you'll see in a database, you'll see a gene, just a bunch of letters spelled out. They just show one of the 2 strands. The complement is not shown because you can figure that out. And it's always, by convention, a database sequence will always start with 5' end, just by convention, just so you know. It was not shown at the complement, right? Everybody's clear on that? Okay. All right. See, in the next lecture, we'll look at a shot of a sequence from a database. Okay. So it's this anti-parallel structure. Okay. The next slide will see this more clearly. The bases, the complement bases are like rungs of a ladder. They're kind of planar. Okay, they're at a right angle to the long axis of the molecule. That enables us to stick a molecule called chlorine bromide and stain it so we can see it. It's right in between the rungs there. Okay, and as this twists, the distance between each base is 0.34 nanometers. There's 10 base pairs in one helical turn, for a distance of 3.4 nanometers. And they knew that from Dr. Franklin's photograph. Okay? And the helical shape is in a right-handed coil. It's like, take your right hand, put your fingers like that. This is the orientation. The helix spins in the right-hand direction. That's called the B form. There are 3 forms of DNA. There's a B, A, and Z. B is the right-handed form that DNA mostly occurs as. Z is a right-handed, but it's more compact. That's what, when we dehydrate DNA, it adopts the did I say Z form? I meant the A form. Okay. B form is the right-handed version that you find in a living cell. That's the most common version. A is right-handed, but it's more compact, and it's usually when you dehydrate DNA, so it's not really a natural form. And there's a left-handed version called Z. And Z does occur sometimes in cells, and it might be a response to viral attack. They tend to notice the Z form when there's been a viral attack. But not much known about it. Mostly it's in this right-handed version called the B form. Okay? And if you look at the outside, you see that just because the helical spin of these 2 complementary strands, there's a minor groove and a major groove. Okay? Alternating minor, major grooves. Now, proteins bind to DNA to do all kinds of things, like secure it in nucleosomes, to control its expression. There are things called transcription factors that bind to DNA to turn genes on and off. And things like restriction enzymes that can cut DNA at certain sequences. These proteins have to be able to determine a particular sequence. So they can turn on a particular gene. They can recognize the base pairs from the major and minor grooves. These proteins can bind, and they can tell the sequence of bases. Okay? By binding to these minor and major groups. So a protein binding the outside can tell this, can read the spelling. There's lots of proteins, transcription factors, restriction enzymes, that need to know where they are to either turn a gene on or off, a particular gene on or off, or a restriction enzyme to cut a particular gene out. We talked about restriction enzymes in this class yet? We have or haven't? We're going to use restriction enzymes next week to cut some of your DNA at a particular location. Okay? It's one of the most important tools in the molecular biology toolkit, discovered in the 1970s. Those enzymes can bind the outside surface, and they know where they are, and they can recognize the bases in these major minor groups. Those anti-parallel, double helix. Okay? The next slide shows you sort of the nature of the geometry a little bit. How the base pairs are really like flattened rungs. If the long axis is vertical, they're perpendicular, like flattened tiles. Okay, you can see 2 hydrogen bonds between A and T, 3 between C and G. We'll talk about the significance of that as well. Okay? Later on. If you think about the structure, how it's suitable for storing information by embedding a sequence of nucleotides, having 2 strands allow for replication. Also, the hydrogen bonds between the 2 strands means it's easy for the cell to unzip the molecule. Okay? Now, proteins can